Okay, good afternoon, everybody. Very warm welcome to this sixth and final webinar in our series, actually, where we've been exploring how the engineering biology mission hubs are harnessing engineering biology to address real-world challenges. So my name's Tom Pearson. I'm a senior portfolio manager for engineering biology at BBSRC. And in a few moments, I'll be handing over to Andy and the P3EB Hub team to take you through all the really exciting work taking place across their consortium. But just before I do that, I wanted to spend a couple of minutes just setting the scene in terms of why we consider engineering biology to be so important at UKRI, and also what we're hoping to achieve through these specific Mission Hub investments. So a small bit of housekeeping to start with. So as this is a webinar, you'll note that your microphone has been muted and your camera is disabled. And we've also disabled the chat function just to ensure that all your questions are captured in the correct place. On questions, you're very much encouraged to submit these to the Hub team throughout the webinar. You can do this through the dedicated Q&A function, and then all your submitted questions will be collated and posted to the panel at the end of the webinar. And finally, we are recording today, and we plan to make the recording available on UKRI's YouTube channel sometime over the next few weeks, so do keep an eye out for that. Okay, so engineering biology is one of our strategic priority areas for investment at UKRI. And the reason for this is because we strongly believe that engineering biology has the potential to deliver huge benefits to the UK in terms of addressing major societal challenges, driving economic growth, and in increasing national security, resilience, and preparedness. So as an emerging technology, engineering biology has the potential to transform a broad range of diverse sectors, spanning agriculture and food systems, manufacturing and clean growth, in net zero and in environmental remediation, and also in biomedicine and in improving human health. And it's really because of this broad range of applications that engineering biology is a technology that's recognised globally to be of major importance, with some predicting that it could contribute alongside other advanced biotechnologies up to $4 trillion annually to the global economy by 2040. So as part of our strategy to support the UK in realising this potential, back in 2024, we invested over £100 million in 6 engineering biology mission hubs and in 22 engineering biology mission awards. These investments were made through UKRI's National Engineering Biology Programme, with co-funding from the wider Technology Missions Fund programme, which is a programme that's provided a substantial amount of investment to grow UK capabilities in the so-called critical technologies. The Mission Hubs and Awards build on the UK's world-class engineering biology capabilities to really drive engineering biology to that next level of technology maturity, unlocking its potential across a broad range of application areas, and ultimately aiming to deliver tangible impacts that make a real difference to how this technology benefits UK society. And now I'm very pleased to introduce one of the Mission Hub project leads, Professor Andy Pickford from University of Portsmouth, who, along with his colleagues that are joining us today, they're going to explain how the P3EB Hub is harnessing engineering biology to address the environmental challenge of plastic pollution. So with that, I shall stop sharing my screen and pass over to you, Andy.
Thank you, Tom. I'll just sort my screen out here. So thank you very much for the introduction. And of course, I should start by thanking UKRI and BBSRC for funding our Mission Hub. So yes, we are the Preventing Plastic Pollution with Engineering Biology, or P3EB Mission Hub, which is a consortium of academics and researchers from 7 universities across the UK. I will give a brief introduction shortly, talking about the background to our mission. I'll then pass over to my colleagues, the majority of whom are early career researchers, to add greater detail onto what we're aiming to do. Artie will talk about public and policy engagement. Angie and Jack will talk about our technologies. And then we'll have Alessia, Dominic, Brooke, and Liz doing a deeper dive into their specific projects on the hub. So first of all, our mission. So plastics are an integral part of our modern society. They provide performance to the clothes that we wear. They protect food and extend food and drink, extending the shelf life and preventing wastage. They protect the medicines that we use and take. They insulate the electronics that we use. They provide light weighting to our transportation systems, our vehicles, and they supply the utilities to our buildings, our homes and our offices. So there's hardly a sector of life that has not seen the benefit of plastics in some way. Now, plastics are very cheap to produce from oil and gas, and they have an amazing variety of material properties. And as such, they're highly desirable. And so production has dramatically increased over the past 50 years from the 1960s and 70s, when production really started to take off. And now we're producing about 400 million tons per annum of plastic. And that's equivalent in mass to about 40,000 Eiffel Towers. So a huge amount. Producing such a large amount of material wouldn't be a problem if we were able to deal with it well at end of life. But unfortunately, our end of life management of plastic is really below where we want it to be. Only about 15% of plastic packaging is collected for recycling at end of life. A similar amount is burnt in some way, either through incineration or in energy recovery facilities. About 40% goes to landfill, where it slowly breaks down into microplastics, which eventually pollute our environment. And about 30% will leak directly to the environment and break down there. Clearly, we need to reduce the amount of plastic we produce. We need to reuse that plastic wherever we can, and we need to recycle better. We need better facilities, we need better technologies in order to increase the amount of material that we can feed back into our plastic system. If we were able to improve plastic recycling technology, we actually impart greater value in the waste. It becomes more desirable to retain it rather than burning it in some way or sending it to landfill. If it has greater value, then it is more likely to be recycled. So we will increase the proportion of recycled material. By doing so, we lower pollution because less is being burnt, and we help to protect the environment because less is leaking directly or indirectly from landfill. Now, mechanical recycling is, of course, well established. It was first used in the 1970s and grew in popularity in the 80s. It is cheap and it's low energy, but unfortunately, it damages polymer chains. So they become shorter through the process, and that means that the material loses some of its desirable properties, and that limits the amount of recycled material that can go into a plastic product. Instead, we can chemically recycle plastics by breaking them back down into their monomers that they originally were made from. We can do this with chemicals, or we can use engineering biology using engineered enzymes to break the bonds linking the monomers together, to purify the resultant monomers away from each other, and then we can either stitch them back together to make plastic polymer chains which are equivalent in properties to the virgin ones, or we can upcycle those monomers into higher value products. And we will hear about that towards the end of the webinar. So our mission then is to support the transition towards a circular plastics economy by using engineering biology to support a system whereby plastic can be broken back down into its monomers at end of life. Those monomers can either be reincorporated into the plastic cycle, or they can be upcycled into higher value chemicals. So those are our first two objectives: to engineer enzymes for efficient plastics deconstruction, and to engineer enzymes and microbes for upcycling into higher value products. Ultimately, we also want to engineer enzymes and microbes for sustainable monomer production, so that we can decouple from oil and gas fossil fuels altogether. We won't be hearing about that aspect of our work today, though. Now, I should say that our technology is designed to complement existing mechanical recycling, because mechanical recycling, which aims to keep the polymer chains intact, will always be lower energy than breaking the polymer and then putting it back together again. So we, in seizing this opportunity, we assembled together 23 academics from 7 universities across the UK. These are our investigators in the top left here. We've recruited a fantastic cohort of researchers, technicians and associated PhD students who are actually delivering the science. And we are steered in our work by a superb advisory board who are helping us to ensure that we produce real-world impact from our work. We're also supported by 10 project partners who are supporting either the technical side of the work or making sure that what we do is able to reach the general public. Now, we're not targeting every single plastic that is made, and there are literally dozens of them, but we're focusing on those that contain carbon oxygen or carbon nitrogen bonds in their backbones. They're shown on the left. There, the graph shows the amount of these that are produced on an annual basis, and you can see it's dominated by PET, which is polyethylene terephthalate. This is the polyester that is used in plastic bottles and food packaging, but it's also the plastic that's in polyester textiles in clothing. Another target of ours is polyurethane, which you'll generally find in foams, mattresses, or these cleaning foams, but is also the material that you find in other textiles that gives it stretchiness. So lycra or elastane is polyurethane. We're also targeting nylons, like nylon 6,6, which you obviously is also used in textiles, but is also very commonly used in ropes. It has very high tensile strength. We're also looking at polycarbonate breakdown, which is polycarbonate is a high impact resistant plastic, which is often used in protective screens, and epoxy resins as well, which have a variety of uses, which you'll hear about later. So we have a very multidisciplinary approach. We cannot cover every single aspect of this today. We're just going to cherry pick a couple. But you can see our expertise maps around the design, build, test, learn cycle of synthetic biology. We actually start at the learn step, because what we do is we take enzymes that are naturally occurring in nature, and we learn about their properties and how they are able to break down plastic to a limited extent. And then we engineer them in iterative cycles to make them more robust and faster and more amenable to industrial deployment. So you'll hear shortly from Angie about the discovery aspects, the enzyme discovery, and you'll hear from Jack about our enzyme engineering approaches. But what's really important is that we are engaged with industry, but also with the general public and with policy, to ensure that what we are doing actually aligns with expectation. And so I'm going to pass over now to Artie Prasad, who is going to talk about our public and policy engagement activity.
Thank you, Andy. As Andy described, we at P3EB aim to open new doors for tertiary recycling processes and new waste-based products that are beyond recycled plastics. So towards the implementation of these sustainable processes and products emerging from our research, our approach is to embed public and policy engagement activities as an integral part of our collaborative work. To do that, we're drawing on the established expertise that we have in our teams at the Plastic Waste Innovation Hub at University College London, the Sustainable Materials Innovation Hub at the University of Manchester, and the Centre for Enzyme Innovation at the University of Portsmouth to integrate public and policy engagement activities throughout. Next slide, please. We recognize that using engineering biology for the molecular repurposing of plastic waste is still an emerging technology. And considering the potential range of products, which include food and medicines that could emerge, we understand that it addresses real-world problems in which public audiences are deeply invested. To us, this means it is essential to foster an open environment of awareness amongst public and policy-making communities to encourage critical and informed analysis, scrutiny, discussion, and debate that's grounded in reliable data. Next slide, please. So P3EB is training our team, and we have a particular eye on preparing our early career researchers to communicate their research to public and policy audiences. We do this through mentoring, through deep dive, skill-based workshops, and by providing strategic opportunities for them to engage with parliamentarians, journalists, public interest groups, and wider public audiences through science festivals and many other outreach events. More broadly, we're working towards understanding and shaping the public discussion on the future and the societal impact of engineering biology's potential by co-developing a joined-up strategy for media, policy, and public communities of interest. Next slide, please. I'm very pleased to pass on to my colleague next, Angie Beckett at the University of Portsmouth.
Thank you. Hello, yes, I'm Angie. I'm a molecular microbiologist based at the University of Portsmouth, and I'm going to speak to you about enzyme discovery. Next slide, please. So we've heard why these enzymes are so important, but now where do we go to find them? Well, we tend to focus our sampling on areas of high plastic pollution, or we may also look in extreme environments such as hydrothermal vents, where it's so warm that the bacteria and the enzymes that have adapted to those high temperatures may have an advantage for use in industry. Next slide, please. Once we've taken those samples and brought them back to the lab, we need to screen them to see if they have some activity against plastic. Now, we might take agar plates and mix them in plastic, and then when we grow the bacteria on the surface, we can see clearance zones if plastic degradation has occurred. We may also measure indirectly plastic breakdown by looking at the byproducts of this process using HPLC. Similarly, we may look at the surface of plastic for evidence of pitting and degradation once exposed to those bacteria, which you can see in this bottom image taken by my colleague, Dr. Jo Herbert. Next slide, please. But finding these bacteria is not enough. We need to understand how they're doing it. And we do this by looking at their DNA. Now, we may take those single types of bacteria that we've grown on those agar plates and look at the DNA that way. But the problem is when we take samples from the environment and we culture them on agar, we lose about 95% because a lot of organisms don't like growing under lab conditions. This means instead we could take a metagenomic approach, whereby we take those environmental samples and we sequence them directly without the culturing and screening step first. Similarly, for a number of years, sequencing experiments have already been taking place on global samples, but perhaps those original researchers weren't looking for the presence of plastic degrading genes. So we can now go back to those databases and look at them retrospectively. Next slide, please. So what do we do with the DNA once we have it? Well, the genes within that DNA give the instructions to the bacteria on how to make an enzyme. So when we look at this DNA sequence, we can make a prediction about what that enzyme structure looks like. If the structure is similar to other known plastic degrading enzymes, then we can make a prediction that the ones we found are likely to break down plastic. Next slide, please. But we need to do more in-depth functional testing. This is especially true for those metagenomic and data mine samples, where we haven't had this opportunity to do that pre-screening step. So for these samples in particular, we take the plastic degrading genes that we found and we cut and paste them into an expression system. This is typically a bacteria that can produce the enzyme in high amounts. Then we want to test that activity in a couple of ways. We might do quick look-see color change assays where we just measure whether or not the enzyme can break down ester bonds associated with plastic. Once we find those that have potential, we may then want to do more in-depth testing, where we purify and characterize those enzymes up against a specific polymer type. In which case, we would measure how quickly and how much of that plastic they're able to break down. Next slide, please. Finally, we may want to better understand the entire process the bacteria use to break down that plastic. It's not always just that initial breakdown of the bonds, but also all the downstream metabolism that happens afterwards. So to do this, instead of looking for the presence of DNA and a single gene, we look at the RNA, and this tells us which genes are actually active in that process. And we do that by growing the bacteria with and without the presence of plastic and see which genes are activated when plastic is present. Next slide, please. Once we've discovered these enzymes, we then need to further enhance them ready for industrial usage. And I'm now going to hand over to my colleague, Dr. Jack Jeffries, who's going to speak to you more on this.
That's great. Thanks, Angie. Hi, everybody. My name is Dr. Jack Jeffries. I'm a lecturer at University College London, and I'm going to talk to you about sort of enzyme engineering. So Andy, if we can have the next slide, please. So as Angie was saying, we go out into the environment and we identify enzymes which are able to break down plastics. And that might be in the sort of numbers of enzymes of tens to hundreds. And those enzymes will have different characteristics. Some of them will accept different plastics. Some of them will operate at different temperatures. Some of them will be more active or less active. Some of them may tolerate various levels of solvent. And these characteristics are different for all the enzymes and may not be the same sort of characteristics we want in our industrial processes, where we tend to run them at higher temperatures and with a little bit of co-solvent, organic solvent to help solubilize the plastic. So these wild-type enzymes are a great place to start, but we like to engineer them and make them better to fit and suit better our needs in that industrial process. Now, as Angie was saying, we have the DNA sequence, and that DNA sequence codes for amino acids. And those amino acids linked together make the protein. And the order and character of those amino acids come together and define the shape and the activity and the substance and temperature tolerance of those enzymes. So by changing that sequence, we can mutate and engineer those enzymes to have those sort of defined characteristics that we're looking for. And those wild type libraries are the best place to start, and we build on that basis and make them suit our needs. So if we can go to the next slide, please, Andy, I'll talk about some of the techniques that we might use to do that. So enzyme engineering approaches roughly fall into sort of two different approaches. One which is a random approach to that mutation, the variation, and one which is a sort of a rational approach. So when you have that DNA sequence, you can make random changes, mutations, which will lead to changes in the amino acid sequence, which will give different characteristics to those enzymes. You can then use the approach Angie was talking about, where you get them into a microbe, or quite often E. coli, to make that protein. You grow it at a large scale and you test it for activity, and you see whether its characteristics have changed. It may have become more active, it may have become more resistant to temperature or solvent. Oftentimes, it will lose activity or become less resistant. You do that random-based approach, you make the enzyme, and you screen for that desired activity. And when you find a good hit, you take that one forward. That's a sort of random-based approach. You can take a more rational-based approach, where you look at the enzyme and you might either have its structure through sort of X-ray crystallography or using some of the new tools like AlphaFold to predict that structure. And you identify amino acids in and around the active site, say, where the plastic will bind. And you say these residues are probably involved with the activity of the enzyme. Let's mutate these ones and see whether we have a gain of function if you're looking to increase activity. If you're looking to increase temperature tolerance, you might look elsewhere outside of the active site, but you're taking more of a rational or a design-based approach. Again, you make the mutation, you produce the protein in a microbial chassis or a host, and then you test and you have to screen for that activity. And Angie's already spoken about sort of zones of clearance. We can see that on the bottom left. You might use colorimetric screens, you can see that on the bottom right, or you'd use sort of more analytical techniques like liquid chromatography, and we can see a sort of liquid chromatogram in the bottom middle. So you make your mutations, you make the protein, and then you screen for that gain of function, and that's that activity screening. If we can go to the next slide, please, Andy. Once we have that ability to make the mutations, what we really do then is sort of hijack the natural process of evolution. Because if you think back to what you know about evolution, this is how it works. You have a mutation, you screen that mutation for fitness. Does it confer an advantage? And if it does confer an advantage, that mutation is maintained. And then that mutation can have mutations added on the top and you gain more fitness. And that's that sort of cycle of directed evolution that we use. You have a sequence, you make your modification, you make the enzyme, you test it. If it's good, you take it back through another cycle where you mutate again, test those further mutants, and test. And you do this cycle of what's called directed evolution. One of the challenges with directed evolution is the number of sequences that you have to generate to find a good or active mutant. And when it comes to random approaches, that can be very high. And even with directed approaches, that can be quite high. So how do we screen those numbers of mutants quickly? We take some approaches in the hub where we use cell-free systems. So we don't use whole cell systems to produce protein. We break them up and we use that liquid to make it, and you can scale that down. But also within the hub, we have the technology to use microfluidics. So that's very small scale liquid devices where enzymes are encapsulated in oil droplets at the micrometer scale. Reactions are run in those droplets and they're monitored. And then we can use very powerful techniques of sorting. And you can see that in the bottom right. The droplet coming in from the top left has a color. That means it's active. As it gets to the divide, you use some electronics to flick that droplet into a channel and you collect those. And those are your active enzymes. And that can get up to testing mutants in the order of tens of thousands to hundreds of thousands of mutants a day. And these are very powerful tools and techniques to really supercharge this mutation process. So if I can go to the next slide. I'm going to introduce now our early career researchers to really talk about the research that they're driving within the hub to discover these enzymes and apply them to these plastics. So I'd like to introduce a researcher who works with here at UCL, Dr. Alessia Tonoli, and she's going to talk about discovering new urethanases.
Thank you, Jack. Hello, everyone. I'm Alessia, and I'm a postdoc researcher based at UCL Biochemical Engineering, working in the Jack Jeffries and Helen Hales groups. Today, I'm going to talk about the enzyme discovery work our group has carried out and is continuing to carry out to identify enzymes for the degradation of polyurethanes, with a particular focus on elastane, also known as spandex, which is a polyurethane-based synthetic fiber widely used in textiles. Next slide, please. As many of you will know, the textile industry contributes significantly to the plastic pollution problem, with a large proportion of waste containing synthetic polymer fibers such as elastane. Elastane is used because of its elasticity and durability, which arise from its alternating rigid and soft segments joined by urethane bonds. Enzymes able to catalyze the cleavage of urethane bonds, also called urethanase, have been reported in the literature, but this is still an emerging field and new enzymes are still required to tackle the sustainable degradation of elastanes. In our group, we approached this challenge using metagenomic mining to search for enzymes capable of hydrolyzing urethane bonds. And we mined one of our in-house metagenomes derived from a shower drain, which was selected as a potential source of robust hydrolytic enzymes. And from this dataset, we focused on identifying urethanases, particularly amidases from the amidase signature family, as promising candidates for elastane degradation. And to support this discovery process, we also developed a high-throughput colorimetric screening assay, which is based on a tyrosinase reaction and allowed us to rapidly test the enzymes candidates. And by combining metagenomic mining with this assay, we identified 3 active urethanases, which were able to break down elastane model substrate first and were then applied to pretreated commercial fabrics. And one of these enzymes enabled the depolymerization of the elastane components. Next slide, please. And building on this work that we recently published, we are now expanding our investigation of the amidase sequence space with the aim of enlarging our toolbox of spandex degrading enzymes and also explore the urethanase diversity. Next slide, please. To address this aim, we have mined additional in-house metagenomic datasets, including samples derived from industrial waste streams and from environments enriched with plastics. And from these datasets, we initially retrieved nearly 12,000 amidase sequences based on interpro annotations, and we then applied filtering steps such as sequence length and other parameters, narrowing the dataset down to more than 5,000 candidate sequences, which are shown here in this sequence similarity network, where each square represents one sequence. And we are currently exploring this sequence space using information both from our previous work and from the literature to guide the selection of candidates to be retrieved, expressed, and tested. With this work that is still in progress, we aim to identify novel and diverse urethanases that could contribute to future strategies for the enzymatic depolymerization of elastane and other PU containing textiles. Thank you very much for your attention. Next slide, please. I'm now going to pass over to Dr. Dominic Wood from the University of Manchester.
Hi, I'm a PDRA in the group of Lucian Wong at the University of Manchester, and our research focus is degrading of thermoset epoxies. Next slide, please. So in contrast to disposable plastics, epoxies are physically and chemically very robust and used typically in long-term applications. And they're ubiquitous in life. So they're used in cars, wind turbines, everything from computers to sports equipment and many, many more applications. And unfortunately, their durability makes them incredibly difficult to recycle. And currently, they're resigned to landfill and incineration. Epoxies are made by mixing a blend of bisphenol-containing resins, like those you can see on the bottom left, and adding a blend of curing agents. And this forms a very complex, heterogeneous cross-linked polymer comprised of lots of strong carbon-carbon bonds, which are very difficult to break. Next slide, please. So we are using oxidative enzymes to cleave these bonds. And because these are heterogeneous polymers, breaking them down produces an incredibly complex mix of degradants. As you can see from this structure here, even if we break only a few types of these bonds, the result is production of potentially thousands of different compounds. And this makes analysis very challenging. So the figure on the bottom half of the screen is a mass spectromatogram of a sample of degraded epoxy polymer. And there's lots of peaks. Within each peak is many coeluting compounds. And because of the sheer number of possible degradants, it's impossible to look at data like this and say, this ion here is probably compound X. Next slide, please. So to address this problem and facilitate better insight into polymer degradation, we are developing chemometric tools and workflows such as the one I'm going to run through quickly here. The first step at the top left takes all the unknown compounds from a degradation reaction, classifies them as being degradants or artifacts. Then some summary statistics are generated to give insight into general trends over the course of the degradation process, like the number of degradants at each time point, and we can do some clustering to visualize the similarity between samples. We then have an algorithm for predicting the chemical formula of degradant. And once we have the chemical formula, we can predict aromaticity. So we can see how many products are monoaromatic or diaromatic, for example, which is something we're interested in. We can also determine whether degradants contain certain chemical motifs. So to do this, we've created a library of MS2 spectra for models of epoxy compounds. And when I was doing that, I found that some of the MS2 fragments are unique to particular motifs, and that means we can use them diagnostically. So, for example, some fragments are only generated from bisphenol A containing compounds. So when we fragment our degradants, if we see these particular fragments, we can conclude that this degradant contains bisphenol A. And finally, the ultimate goal is to predict the complete structure of degradants, and that's work that's in progress. Next slide, please. So that's some of the work that we do, and I want to quickly touch on why we're interested in the product from polymer degradation. So one reason is to identify and monitor toxic byproducts. Another is to identify degradants that can be upcycled into high-value products, which you'll hear more about shortly from Liz. And finally, we heard earlier about how valuable screening methods are for finding novel plastic-degrading bacteria and enzymes, and also for then optimizing the plastic degradation process. And if we can analyze our samples and identify degradants, we know what's being produced from the breakdown of plastics, then we can facilitate the development of these kind of high throughput screens. I've just got two examples here. So on the left, we found out from our analysis that we get phenolic degradants from degradation of epoxies. So we employ a colorimetric assay that can detect a range of phenols, and we can use that to optimize the reaction for increased degradation in general. And on the right, we also identified hydroxybenzaldehyde as a specific product of degradation. And we've got an assay that will detect that, which we can then use to drive the degradation process towards producing that particular compound. Next slide, please. So that's a snapshot of the work that I'm doing. Thank you for listening, and I'll now hand you over to Brooke.
Hello, I'm Brooke, and I'm a postdoctoral researcher based at Imperial, working in the Jose Jimenez lab, engineering microbes as platforms for plastic hydrolysis. Next slide, please. Across the field, the most widely adopted approaches to polyester plastic biodegradation rely on enzymes that are extracted and purified from microorganisms, as you've heard. And these purified proteins are then applied in a cell-free system to demonstrate and study plastic hydrolysis. But in most cases, the system stops at the enzyme, and continued activity depends on repeated protein production and purification. Next slide, please. So what's largely missing is a living regenerative system, one in which the organism itself produces the enzyme and the protein, but can also assimilate the plastic products post-hydrolysis. So our work at Imperial is to generate organisms that produce plastic degrading enzymes, release them to access the polymer, and then take up the resulting monomers and convert them into biomass. With that biomass growth, then supporting further enzyme production, allowing it to grow on those breakdown products, so turning the waste into biomass, or into new value-added products, also known as upcycling, as you've heard, which we further discussed by my colleague Liz next. Next slide, please. So to do this, we start from utilisation rather than degradation, where we first identify microbes that already have the metabolic capacity to grow on plastic-derived monomers. This can be introduced or enhanced through various approaches. Environments may already have relevant metabolic pathways in place. Now, using this approach, we have identified multiple bacterial strains capable of growth on PET-derived monomers in pure culture and have combined growth characterization with genomic analysis, just to confirm that they actually do have the presence of those catabolic pathways. Now, from this process, we've isolated several Pseudomonas strains, which are our ideal candidates at the moment. And we further engineer these organisms to produce value-added compounds such as violacin, while also using the terephthalic acid, that byproduct of PET hydrolysis, as you can see in the top, that violet colour. Now, once we have these microbes that can already assimilate the monomers and use the kind of the TPA or the ethylene glycol, the next step is to engineer access to the polymer itself. We do this by introducing plastic degrading enzymes, enzyme expression systems into these strains with a particular focus on the efficient extracellular secretion. Now, for polymeric substrates like PET, secretion is obviously essential because the enzyme has to reach that plastic before any utilisation can occur. Next slide, please. So to achieve this, we have taken advantage of the native type 2 secretion machinery in Pseudomonas, where we have fused candidate proteins to a signal peptide, enabling the proteins to be exported in a folded state via the TAP pathway, as you can see in the top left. And this enables the extracellular secretion. And we screen multiple proteins, as you can see on the top right. And this allows us to see different halo formations, just as a qualitative way of looking at it, but we also do lots of quantitative analysis. Next slide, please. Now, when we introduced various proteins, we found that PHL7, which is quite a well-known protein, was most optimal for our system. And then we have introduced that into our Pseudomonas strains, where we observe growth in the presence of PET compared to a control where you don't have that PET, that plastic. Now, over longer incubations, you can see in the bottom right graph, these strains actually show sustained growth. Now, this indicates that that enzyme production, the plastic hydrolysis, the metabolism, they're all functionally coupled, which creates this regenerative loop. Next slide, please. All in all, this work pushes microbes towards becoming both a catalyst but also the consumer. And at this stage, the key question is no longer whether or not these microbes can perform these functions, but it's actually how fast and how robustly can we engineer them to do, so whether or not we can optimize them to the level that we need. As highlighted here, these systems are functional, but it's their performance which is not yet comparable to the purified proteins themselves, and it's not hitting that industrial kind of viability that we need it to. So there's many areas where we have active research, including kind of increasing extracellular enzyme yields, so maximising that production, also balancing that growth with enzyme production. So now, while our current focus is on controlled growth coupled systems, these same principles actually also apply to bioremediation. For example, in the cases of uncollected plastics such as microplastics, microbial processes could be used in engineered settings such as wastewater treatment plants to kind of contribute to that pollutant removal. Now, finally, this kind of platform opens up a lot of opportunities for plastic upcycling, whereas plastic carbon cannot be just transformed into biomass, but also into high-value products, which links nicely to our next talk. Next slide, please, where I can introduce Dr. Liz Lau from Edinburgh, who will also show how microbial systems can be utilised and engineered towards value-added product formation. Thank you.
Hi, thanks Brooke for that. So my name is Liz and I am a postdoctoral research associate at the University of Edinburgh in the Joanna Sadler Lab, and today I'll be talking about harnessing microbial and biocompatible chemistry for plastic upcycling. Next slide, please. So harnessing microbial chemistry can be done in two ways. First of all, by fermentation. And this is when a waste feedstock is broken down into a fermentable monomer and then is fed into an engineered microbial organism. And the fermentable monomer grows through the metabolism of this engineered microbe and can be used as both as a growth source and for the conversion to value-added chemicals. The second way is to use whole-cell biocatalysis, and this is where an engineered microorganism is used at a high optical density and is fed a renewable feedstock, and at the same time a waste feedstock as well. And this is then fed into the process, and this engineered microbe should then convert the waste feedstock again into value-added products. So next slide, please. So currently, plastic waste is emerging as a novel waste feedstock, and here I'm kind of showing all the different types of plastic waste that have been chemically or biologically depolymerized and converted to value-added products. So next slide, please. So I'm going to hone in on kind of these three quite important chemicals that have been, or the products of interest that have been highlighted here. And so currently we have made vanillin from pet waste, and this is commonly used as a fragrance and flavour compound. We've also been able to make adipic acid from pet waste, which is a precursor for nylon and used in many garments or clothes that you wear. And lastly, we have acetone which is most commonly used as nail polish remover, and that is from the plastic PHB. But as you can see, as extensive as this scope is, it is still limited, and we want to be able to develop a technology that can expand this scope of products and create even more products that we already have access to. So next slide, please. So here we are proposing that by exploiting the metabolism of microbes and the non-enzymatic reactions that have been provided by chemistry, we create a space that is biocompatible chemistry, where a waste feedstock like plastic can be depolymerized into its monomeric substituents and thus be fed into the engineered microbes and create an intermediary platform. And then couple that with biocompatible chemistry, we can then expand that product scope and hopefully make even more products than we already can from waste. So next slide, please. So what I'm currently working on with my research, it's focusing on the depolymerization of PET plastic into terephthalic acid, which is its monomeric form, and then using an engineered E. coli to convert this to vanillin, which is our intermediary platform, as I'd said before. And then by combining it with biocompatible chemistry, we can make zingerone, which is this incredible flavor and fragrance compound that has a smell reminiscent of ginger and cloves, and is currently worth roughly around £350 per kilogram. So hopefully by using biocompatible chemistry for sustainable chemical production, we can use this technology to close that loop and create a more circular economy in this way. So thank you for listening, and now I'm going to hand back to Andy for closing remarks.
Thank you, Liz. Hopefully that has given everybody a clearer view of what our Mission Hub is about and what we're aiming to do and the progress we're making to date. I just want to thank everybody involved in the Hub. As you can see, it's a very long list of investigators, of researchers and technicians and associated PhD students. And I'm grateful to every one of them for the way that they have embraced the mission that we are undertaking. I really want to say again a big thank you to our advisory board. It's Adrian, Gorg, Kate, Sally and Maria for the direction that they've provided for us. That's been absolutely crucial to make sure that we're going to deliver some real world impact. And also to our project partners who have been fantastic in supporting these efforts. And finally, of course, I want to thank UKRI and BBSSC for the funding, because none of this would happen without it. And I'd like to thank the whole UKRI engineering biology team for the support that they have provided to us for the last couple of years. So that's it from P3EB. If you want to know more, you can visit our website at p3eb.org.uk, or please do email us at contact@p3eb.org, or please ask any questions you may have now. Thank you very much for listening. We are prepared to take any questions and hopefully provide answers for you.
Okay, brilliant. Thanks very much to P3EB Hub colleagues for an excellent presentation. So as Andy said, we can move into the Q&A session now. Andy, if I could ask you to stop sharing your screen, if possible. Excellent. And we do have a few questions that have come in. So the first one is, what is biocompatible chemistry?
That's one for Liz, I think.
I think that is. So basically, biocompatible chemistry is any type of non-enzymatic reaction that can be coupled with the microbes in one pot that can basically do the chemical reaction. Hope that's a good enough answer.
Thank you very much, Liz. Okay, so the next question is, from a risk perspective, how do you evaluate whether microbial degradation reduces overall hazard?
So yeah, so I will pass that maybe to Brooke to answer this one in a second, but I should add that when we're using purified enzymes, which is probably the larger part of our work. We're doing that in contained environments because we want to recover the products, the monomers, which we can then recycle or we can upcycle. But yes, there is the option of using microbes for this. They could also be done in a contained setting, but I guess also you can imagine deploying these in some way. But Brooke, could you take the question from me now?
Yeah, so for us as well, using microbes is still within a contained system, but if it was to be engineered to be external, there's many fail-safes themselves that can be engineered into organisms, which can create auto kind of kill switches, or we have specific targets where, because these organisms can be quite fussy, or the proteins they're producing are very specific to different conditions, most times they can actually be engineered.
I think we've just lost Brooke there, unfortunately, so perhaps we can come back to that question if Brooke has anything else to add.
Into different organisms.
Apologies, Brooke, you just broke up there a little bit.
Oh, no worries. I was only going to say about a lot of things we're doing are based on nature, so a lot of the natural organisms we are introducing, let's say into a soil kind of microbiome, there'll be extensive testing beforehand anyway, but we're not introducing anything too hazardous if you were to anyway, and there'll be many characterizations before that would happen.
Great, thanks, Brooke. Okay, so the next question in from Victoria Boys. So this question is, do the resulting monomers retain their polymerizability, i.e. are CC bonds re-established?
In the case of PET, then we're breaking ester bonds. So in fact, most of our targets are all linked via either ester or amide bonds. They're all condensation polymers. So when you hydrolyze those, you generate acids or alcohols. In the case of polyesters, you generate amine groups and carboxylates in the case of nylons. Polyurethanes are a little more challenging. And perhaps, can I pass to you, Jack, to talk about polyurethanes?
Well, thanks, Andy. Yeah, we're still in early stages, but we haven't yet attempted to repolymerize up to polyurethanes. We're taking more of a track where we're doing valorization to higher value-added products, potentially to pharmaceuticals. I can see there's a question coming up later. But at the moment, we're still early stages. A lot of work has been done in PET, in the area of PET, and that's much more advanced. Polyurethane's still new. We're still discovering those enzymes. So at the moment, we're just finding enzymes which break them down, break at that carbamate and not the ester, because that provides a bit of a more valuable molecule. And then we take it from there. We have to do a series of extra enzymatic or chemical steps before we can get something which might be of value. So we haven't yet tried to repolymerize up to a spandex-like molecule. But I mean, theoretically, as with PET, once you've gotten down to their basic monomers, it does require some chemical treatment and some clever reaction design. But yeah, they're no different then from their, oil-based analogues or derivatives of the same chemical.
Excellent, thank you. So the next question we have is, hydrothermal vents have hopefully not seen any plastic. So how much luck have you had mining them?
I think that's probably a question for Angie.
Yes. So, well, the answer is actually they have seen plastic, unfortunately. So there have been studies where we've, not within our research groups, but other groups globally, where they've looked at the level of plastic in all the ocean layers and they found them deep in the benthic zone. So they actually are in those areas. However, something else to consider is a lot of the activity we have against plastic is from the enzyme promiscuity. So it happens to be able to break down plant material. It's designed for breaking down plant material. And then it just so happens that it can also break down plastic because the chemical structure is similar. So it may not be that it needs to be exposed to plastic in order to adapt to be able to break it down later on.
Thank you. Next question. So when you produce pharmaceuticals from waste, do regulatory bodies raise concerns?
Liz, I think that's one for you. Sorry.
Yes, is I guess the short answer to that. Yes, there probably would be some concerns, but don't forget, I guess a lot of the research that we do at the moment is to prove, and it's a proof of concept that we can do it. Obviously, we would like, you know, as you scale up, you would obviously employ the correct processing and purification techniques that you would normally have in like even in the pharmaceutical industry when they make their pharmaceuticals from oil and gas. So it's just a matter of finding a different way of approaching that. But obviously, currently at the moment, these are kind of those proof of concepts. But I suspect in the future, yes, it will be a concern.
Could I just ask Artie to add to that from a policy angle?
Yes, I mean, as it's proof of concepts at this stage, but as it progresses, it will have to go through the normal checks and balances to make sure it's safe for human consumption. And also in a public sense, this is why, I mean, you know, government funding is behind this and backing these our explorations, but it has been shown time and time again that when new technologies like this come out, they do rub up against particularly the media and the messages they sent out. And that's why we want to work so closely with public audiences and stakeholders to make sure that the messages we're sending across are based on robust evidence and to explain very clearly in context the results we're getting and why if they are released for use, they will be tested and safe to do so.
Thank you very much.
Thank you.
So the next question we have here. So you mentioned that mechanical breakdown is more economic, but in the long run damages the fibre structure. Are there types of plastics more suited for mechanical breakdown, and should their use be encouraged?
That's a good question. There are some plastics are less susceptible to chain breakage than others. PET is susceptible to chain breakage, particularly if it's not thoroughly dried before it goes to a mechanical process, and that's because of this hydrolysis, as we mentioned, of the linkages between the monomers, and any moisture in the air can contribute to hydrolysis of the chains. So other plastics called polyolefins, these are these ones which have pure carbon-carbon backbones, are more resistant. And you'll be aware, for example, of milk cartons, which are made of HDPE, that's high density polyethylene. These are mechanically recycled very effectively. The problem, of course, with those type of plastics is they're incredibly difficult for natural processes to break down, because we've effectively engineered indestructible materials, whereas polyesters do have this kind of Achilles heel of the ester bond, which can be broken, as we know, by the kind of natural enzymes which Angie and others are discovering. So polyesters really are a good compromise between the material characteristics that we want, but also having that potential circularity, where we're obviously not at the point yet where we can say polyesters are truly circular. But that's, I think, it should be the focus of efforts going forwards, is to design polyesters that are, well, recyclable by design. So new types of polyester, not necessarily PET, but other types of polyesters like PLA that you've heard about, and others. So yes, that's a long-winded question, long-winded answer to a very good question. Certain plastics are more susceptible to damage through mechanical recycling than others, but you have this trade-off between the potential damage that they will then cause if they escape to the environment.
Okay, thank you very much, Andy. So we are at time now, unfortunately. So happy to draw it to a close soon. But if I could just ask one final question, and you mentioned it at the end there, Andy, but what is the best way for interested parties to get in touch with you all at the Hub?
The best way is by email address, the contact at P3EB that was on the final page. Let me just make sure I've got the right one. Contact at p3eb.org. But if you just do a web search for P3EB, you'll find our website, and you'll find a link there to the contact email. So probably visiting our website is probably the first point of call.
Brilliant. Okay, well, with that, just a final thank you to all of the presenters today. Thank you to the audience. And as I said at the start, the recording of the webinar should be live on UKRI's YouTube channel sometime over the next few weeks. So do keep an eye out for that. And thank you very much to everybody.
Thank you, Tom.
Thank you. Bye-bye.
Bye-bye.